Welcome to this podcast from Neurogastroenterology Motility. It publishes original research and topical reviews on basic and clinical aspects of gastrointestinal sensation and motility, as well as brain-gut interactions.
Welcome everyone to this month's podcast from Neurogastroenterology Motility. My name is Adam Farmer and I'm a gastroenterologist at the Wingate Institute in London. This month it's my real great pleasure to welcome Dr. Alan Bradshaw. Alan is a research assistant professor of physics and biomedical engineering at Vanderbilt University. Alan, many thanks for joining us on the podcast and congratulations to you and your co-authors on your paper entitled Diabetic Gastroparesis Alters the Biomagnetic Signature of the Gastric Slow Wave. So Alan, could you provide me some background to the electrical activity of the stomach and what is its relevance in gastroparesis?
So the stomach's ability to mix and empty relies on underlying electrical activity in the cellular syncytium of the smooth muscle and enteric nerves and interstitial cells of Cajal all work in conjunction. Typically, the syncytium is well coupled and produces a rhythmic slow wave that propagates down the longitudinal axis of the stomach. Gastroparesis is literally paralyzed stomach, prevents normal emptying. Several studies have suggested that gastroparesis is associated with the loss of ICCs and therefore disruptions in the electrical syncytium. And so our study investigated the electrical parameters associated with pacing and propagation of the gastric slow wave. Structural imaging with things like CT and ultrasound can determine gross anatomical etiologies, but we're looking at functions. The functional disorders like gastroparesis can occur without any evident structural abnormalities.
So perhaps for some of our fellows who are listening to the podcast this month, could you just give us a summary of what electrogastrography is and how it's recorded and measured and interpreted?
Sure. So the electrogastrogram is the recording of the gastric slow wave using electrodes placed on the cutaneous surface on the abdomen. The slow wave is recorded by EGG electrodes as the summed electrical potential of the cellular syncytium at the surface of the abdomen. Historically, EGG is recorded with only a few electrode leads or even one or two. But recently, people have started looking at multichannel EGG. The problem with cutaneous electrodes has been that intervening abdominal tissues tend to smooth and attenuate the electrical potential as volume conduction occurs. So spatial information like propagation characteristics are not measured reliably.
I think that's certainly been the experience when we've used it in the research environment as well. So taking that on a little bit further, could you give us some background to the entity of magnetogastrography?
Yeah, sure. So as I said, electrical potentials can be measured with appropriate electrodes, but there are also bioelectric currents associated with the slow wave. And if you want to measure these currents non-invasively, we look at the magnetic fields they produce. As I said, the electrical potentials are affected by alternating low and high conductivities of abdominal tissues, but magnetic fields are affected primarily by magnetic permeability of the tissues. And for most abdominal tissue layers, the magnetic permeability is roughly the same as the permeability of just air. So the magnetic field may be a more direct reflection of the bioelectric currents from the slow wave, but it's ridiculously small. You know, magnetic fields from bioelectric currents are generally on the order of picotesla, 10 to the minus 12 Tesla. And just to give some perspective, the geomagnetic field of the Earth, which we must mitigate in our recordings, is on the order of microtesla. So we're measuring a signal that's 6 orders of magnitude weaker than the Earth's magnetic field. If you want to do that, you've got to use Currently, you have to use a Squid magnetometer, a superconducting quantum interference device that uses quantum principles. And since it is able to measure magnetic fields as weak as one flux quantum, squids have been called the world's most sensitive detectors of anything.
So I'm right in thinking, in summarising that point, that essentially with the magneto technique, you can essentially get rid of the noise that the intervening tissues between the stomach and the outside world provide. Is that a correct summary?
Yeah, you still have to deal with secondary source effects from discontinuities in the tissues, but the idea is that those effects are not as severe as with the electrical signals.
So what were the methods that you used in your study, and what was your study population?
Sure. So our hypothesis was that non-invasive multi-channel EGG will reflect the spatiotemporal changes in the gastric slow wave that you see in patients with gastroparesis. And we hypothesized, in particular, that EGG and MGG both would detect changes in temporal slow wave characteristics like frequency and percent power distribution, but multi-channel MGG, which is not as smooth and attenuated, would also allow us to evaluate slow wave propagation. And so we suspected that changes in the coupling of the gastric syncytium associated with gastroparesis would disrupt propagation as measured by the MGG. As I said, we use a Squid magnetometer. The magnetometer is 19 sensors with that are arranged sort of in a honeycomb pattern around roughly the area of the circle with a 10 centimeter radius and placed outside the abdomen. The Squid, the way that the Squid works is simply to convert magnetic fields into voltage signals that you can then record on a PC. And so then we analyze the MGG and EGG signals for dominant frequency, percentage of power distributed in Brady, normo, and tachygastric frequency ranges. And then we look at multi-channel analysis using most recently a blind source separation technique called SOBI, second order blind identification. It allows us to identify specific signal components and eliminate specific noise components in multi-channel signals. And then you can take the SOBI reduced MGG or EGG and look at the spatiotemporal progression to identify propagation patterns. We did the same thing. Well, with most of our EGG studies, we use the standard 4-channel approach that other investigators have used. But in the last 2 studies that we did, we expanded our EGG array to 16 channels with a 4x4 grid to see what we could see with multi-channel EGG.
And I'm right in thinking your study population was patients with diabetic gastroparesis.
Yeah, that's correct. It was our study population was all female, 7 females, and then we compared with 7 healthy controls that were both males and females. But I think it's one of the interesting questions that's still open is why this disease affects predominantly females over males in something like a 4-to-1 ratio.
So what were your key results that emanated from your study?
Yeah, so we found the dominant slow wave frequency is not sufficient to distinguish gastroparesis patients from controls for either EGG or MGG. We didn't find any significant differences in percent power distribution from EGG signals. In the MGG, we found that patients had larger percentages of slow wave signal energy in Brady and tachygastric frequency ranges, which may indicate uncoupling. We also showed that the slow wave propagation patterns measured in the MGG were retrograde or static in patients, whereas healthy controls show anterograde propagation. Propagation velocities we computed from our spatiotemporal maps in the MGG were significantly different in patients. Most of them were negative. They were propagating retrograde, and there was a much greater variation in the propagation velocities. And then when we looked at propagation in the 216 channel EGGs that we did, we found evidence of global propagation, but the phase shifts tended to be very abrupt. But it at least encourages us to continue looking at multi-channel EGG processed with something like SOBI to determine whether we can mitigate the effects of volume conduction sufficiently to be able to assess propagation in multi-channel EGG as well as MGG.
So what were the limitations of your study, do you think?
Well, we had a relatively small subject population, only 7 patients. And so that makes it difficult to stratify patients according to the severity of disease, or control of diabetes would be another interesting thing to look at. And we'd certainly like to know how these things affect slow wave parameters. Currently, the MGG is limited to recording with a Squid magnetometer. The fields are just that weak. And these studies have to be done in a magnetically shielded facility. So cost and availability is one potential limitation of MGG compared with EGG. Of course, if the MGG provides critical diagnostic information, then that would certainly help to alleviate some of those concerns. I'm sorry.
Do you think some of the differences that you observed in either the propagation velocities or the waveform patterns might allow clinicians to begin to stratify their pharmacotherapeutics in patients with diabetic gastroparesis in the future? Because certainly in my experience, these patients are often very difficult to treat.
Yeah, absolutely. I think that's one of the big open questions is, exactly how do these propagation patterns respond with different disease etiologies? If we could, there have been some good serosal electrode mapping studies that have suggested that these patterns may change, but we need to be able to correlate those with these non-invasive EGG and MGG measurements. If we had a more complete understanding of the pathophysiology, that would certainly facilitate interventions during therapeutic plasticity. We only looked at diabetic gastroparesis, but it seems clear that gastric emptying alone is not sufficient to separate even diabetic from idiopathic or post-surgical gastroparesis. And so, the use of multi-channel EGG or MGG to non-invasively assess the effects of gastroparesis on slow wave propagation could show differences and provide quick diagnosis and early treatment potentially.
And I agree. I think certainly in my experience, patients the severity and burdensomeness of their symptoms can be very variable. And not always associated with prolongation of their gastric emptying time. So where do you think the knowledge gaps lie in the field, and how do we go about resolving these in the future?
Yeah, I think technologically it's still an open question to what extent the slow wave electrical activity truly reflects pathology. And precisely how mechanical activity influences EGG and MGG. We need better gold standards for validation, like serosal mapping. How does the multi-channel EGG reflect underlying slow waves? I think is a big open question. And what are the specific advantages of EGG and MGG? If you can mine the EGG and get most or all of the same information that MGG gives, then the lower cost makes it a very attractive alternative. Seems clear from our studies and from other labs that frequency dynamics alone are really not sufficient even to distinguish gastroparetics from healthy controls, let alone to stratify them by disease severity. So it would appear that multi-channel spatiotemporal characterization of functional abnormalities is really essential. On the clinical front, I think it's a good question as to how correlated slow wave aberrations are with functional gastric disease, and are there specific indicators that would predict which patients would benefit from particular interventions like medical therapy or pacemakers. And then can these methods be used to better distinguish gastroparesis from other functional GI disorders and to stratify different varieties of gastroparesis? I believe most researchers now think of gastroparesis on a spectrum of functional GI disorders, but relatively little diagnostic criteria seem to be available that separates these disorders and direct treatment regimens. So I think that may be where EGG and MGG have a part to play.
So that's super, Alan. So thank you very much for that. And with that, I'd like to thank you and your other co-authors for a really excellent paper and for assisting in this month's podcast, but also to our listeners for tuning in, and look forward to welcoming you again on another instalment of the podcast next month.
Further information about this paper can be found on the journal website. We hope that you have enjoyed this podcast, and we look forward to welcoming you.